helper T cells, and there's what we call the rule of eights that I'm going to mention right now, which is helper T cells recognizes things displayed on MHC2. So here's a rule of eights. Which is 1, right? Helper T cells, also known as CD4 cells. CD4 count is what we use as a measure to see if you have active HIV. CD4 is a receptor specific for helper T cells. The helper T cells react with MHC2 receptor. 4 times 2, 8. What does it allow us to remember? Which cells react with which receptors, right? CD4, helper T, reacts with MHC2 receptors. What's the other rule of 8 then? The next category of cells, cytotoxic T cells. Cytotoxic T cells, TC cells, are called Cd8 cells. Those cells react with MHC1 receptor. 8 times 1, 8. So we know that cytotoxic T cell will then react with cells that display MHC1 proteins. Right? Again, MHC1 receptors will display proteins from your host, from your body. If it gets infected, it's going to start displaying different proteins than it should. That's when we can find out. Or that's when it's okay to start killing that virally infected cell. So let's talk about cytotoxic T cells now. With cytotoxic T cells, we're going to see cytotoxic T cells. Very important to actually do the killing, right? The weird thing is, helper T cells are incredibly important, but helper T cells do not do any killing. Their main job is to be in the background, helping everything activate. That's all helper T cells do. But we're going to see that's an incredibly important task. When helper T cell population decreases, like in HIV, you are not going to activate these cytotoxic T cells. You're not going to activate your B cells. In other words, your whole specific immunity shuts down without helper T cells. Even though it doesn't do any killing by itself, it will help other cells that does. So let's talk about the cytotoxic T cells. Right? Cytotoxic T cells, part of cell-mediated immunity, most effective against intracellular microorganisms, viruses, fungi, even intracellular bacterium, right? And parasites like malaria that are intracellular. So we need this cell-mediated immunity to fight off things that infect our cells. And their main job here is that you have helper T cells that helps us activate our specific immunity. And as part of that, we have the second class of T cells called cytotoxic T cells. That's our Cd8 cells. So Cd means it's a name for a receptor found on these, right? For cytotoxic T cell, the receptor is called the Cd8, clusters of differentiation. That's what the CD stands for. It allows you to differentiate one T cell from the next. These cytotoxic T cells function in 2 ways. They kill virally infected cells. So these are the ones that actually do the killing, right? Helper T cells are in the background. They help activate the cytotoxic T cells. But once active, it is these cytotoxic T cells that will then kill virally infected cells. It lyses virally infected cells, tumor cells, and tissue transplanted cells. In other words, they're not all good, right? So if you have an organ transplant, unless you have an identical twin laying around, you will have some differences. Even brothers and sisters, even fraternal twins are going to have right? Very different genome from one to the other. So they're going to have very different type of proteins they display. What happens is these cells, cytotoxic T cells, are going to be the one that kills those organ transplanted cells. Major lysin is performed, which punctures a hole in the plasma membrane of the target cell. They also produce cytokines, which promote phagocytosis and inflammation. It's part of our antibody-mediated immunity as well, right? Memory cells are produced, let me phrase that, right? As in antibody-mediated immunity, memory T cells are also seen and saved. And we're going to see that sometimes we're going to have a kind of a reaction called a delayed type hypersensitivity reaction. And we're going to see it in certain times of our, right? Usually the best example of this kind of hypersensitivity or allergic reaction is when you've been exposed to TB and you are given the TB skin test. And we'll talk about that at the end of class. So let's talk about this, and then we'll take a break. What is cytotoxic T cell? So before, we talked about how each helper T cell has a specific receptor that recognizes specific epitope. Same thing with the cytotoxic T cell. Now take a look. The cytotoxic T cell here has a specific receptor, and what we're going to see is that that receptor will only react and bind when it has a displayed epitope that matches. Now the weird thing is this. Usually the epitope that matches is found on a virally infected cell. What do I mean? Let me get as many colors as I can here. So what I mean is this. When you have so right here you have a target cell, it's displaying a process antigen. In a second, we're going to make sense of what that means right here. So with helper T cells, notice they're in the background, helping out, right? The only thing they do to help out is to release interleukin-2. That helps activate. But as part of activation of the cytotoxic T cell, you need to have cell recognition first. So what happens? Let's say you got sick, right? You have these simple cuboidal cells in your bronchioles, because you do have simple cuboidal cells in the bronchioles. Normally, they're going to be displaying MHC1 receptor, and this MHC1 receptor will have my protein on it. So all these cells are going to have the epitope for me. Different epitopes on its MHC1. Now, what happens when I have a virus? I have the varicella zoster virus coming in. And it's got all those different epitopes, including epitope A, like we saw before. Right? What happens? The virus gets into the cell. As the virus goes into the cell, unlike with macrophages, in macrophages, we can kill that cell, right? We can kill that virus in a macrophage. But in this example, there's no killing. It's actually infecting our cell. So the virus gets into our cell. The virus then goes into our nucleus, and it's going to put a little bit of its DNA into my DNA. As it does so, my DNA polymerase makes copies of it. Right? What else? I also undergo, right, transcription and translation. And I start making proteins for the virus. Because you need a capsid. In order to make the capsid, you need proteins. That happens because of translation and transcription. When that happens, I start making viral proteins. The viral proteins start being made and shifted. And now I'm going to display a different epitope on my MHC1. Since I'm making viral proteins, I'm going to start producing the viral protein and sticking it on my MHC1 now. What happens? There's a change in the protein that I'm presenting now. Right? This cell, uninfected yet, is displaying my normal host, right, antigen or epitope. This one has been infected. In the process of getting infected, it's taken over our machinery. It has reproduced its own capsid. It's reproduced its own genome by taking over our machinery. In the process of doing all that, it's going to need to make a lot of proteins. And I'm going to put the viral protein into my MHC1 receptor. MHC1 receptors only display proteins made by the cell. So now I'm making proteins for the virus made by my cell. What's going to happen? That viral protein now gets displayed on that MHC1. All right, what makes it good is that I have this cytotoxic T cell ready to go. The cytotoxic T cell right here has a T cell receptor that recognizes epitope A. Now, this T cell receptor, when it goes to this cell, doesn't recognize that epitope, right? Or it does, it recognizes and leaves because it's a normal host protein. But once it goes over here, and now there's recognition, now it makes it okay to kill the cell. And not only that, we need to kill the cell. If we don't kill this virally infected cell, you're going to produce billions of viruses from this one cell. So you need to kill that cell. How do we kill it? We kill it because the epitope it displays now matches the receptor. Should it match the receptor if it's not the epitope that's from the virus? No, right? MHC1 should normally just present your host protein, my protein, right? But now it's been infected, it's producing the virus's protein. Because it's producing the virus's protein, I display the virus's protein on my surface. Now, I can recognize it. I can attach. I can have perforins and kill the cell, right? So you need that recognition. Once you have that recognition, then you have another helper T cell here. The helper T cell's main purpose is just then to release interleukin 2. Once interleukin 2 is produced, this helper T cell, right, producing this interleukin 2, the interleukin 2 binds to its receptor on the cytotoxic T cell, and now I can activate the cell. Right? So you need that cell activation, and it's a two-step process. First step process is to have cell recognition. There's a T cell receptor. It displays that now mutated form of MHC1 that's now displaying a foreign epitope. You should not do that if it's not infected. And now there's recognition. Now we have other co-stimulants kind of binding it together. The helper T cell releases interleukin 2. The combination of recognition and interleukin 2 stimulates this cytotoxic T cell. Now the cytotoxic T cell forms clones of itself. One of those clones is going to go around and kill any cell that displays that epitope. So then we can kill the virally infected cells to decrease the amount of virus that's floating around. Perfect. All right, let's take a little break because I know your heads are probably